Good afternoon, good morning, and good evening to everyone from all over the world. My name is Adam, and I'm the Field Application Scientist at Twist Bioscience. Today, I'm really delighted to welcome all of you to the Twist webinar, Precision with a Purpose, Benchmark 659 for Cancer Care, powered by Twist Bioscience. So in this webinar, you will hear from our distinguished speaker, Dr. Vivek Rathi and Dr. Lynn Fink from LifeStrands Genomics Australia, on how Twist target enrichment technology and custom panel solutions can enable the laboratories to bring improved clarity and confidence to their cancer research. So now I would like to formally introduce our speakers, Dr. Vivek Rathi and Dr. Lynn Fink. So Dr. Vivek Rathi is a molecular and anatomical pathologist, is the medical director of LifeStrands Genomics Australia, a NADA and CAP accredited genomic laboratory providing high-quality, affordable, and rapid molecular testing for cancer patients. A fellow of the Royal College of Pathologists of Australasia and the RCPA, and with expertise in translational genomics and clinical implementation. He leads a multidisciplinary team committed to innovation, accuracy, and patient-centered care. Previously, Dr. Rathi was head of genomics at Monash Health and has also served as a staff specialist at St. Vincent Hospital in Melbourne. We also have Dr. Lynn Fink, a senior clinical sciences specialist in clinical bioinformatics. She is the laboratory director of LifeStrands Genomics Australia, a fellow of the RCPA Faculty of Science and Pathology Informatics. She has expertise in leading clinical molecular pathology laboratory teams and in translational genomic science. So without further ado, please welcome Dr. Vivek to start his presentation.
Hello, everyone. Welcome to this webinar where I and Lynn will be sharing with you some salient features of our newly launched comprehensive genomic profiling NGS assay called Benchmark 659. The agenda outlines the key topics we will cover today, starting with an introduction to LifeStrands Genomics Australia. We will discuss the rationale behind creating Benchmark 659, its features, and provide case studies to illustrate its applications. We will also delve into the technical details and some validation data for this panel as well. So LifeStrands Genomics Australia, or LSGA in short, is a genomic lab located in Mount Waverley, Melbourne, Australia, and is fully dedicated to cancer genomics. We are a NATA accredited lab operating since 2022, and we are the only CAP accredited lab in Australia. Our operations is governed by Australian laws and fully compliant with NATA and RCP and NPAC guidelines. So here I've shared with you the LifeStrands Genomics test portfolio. We also offer some other PCR-based tests like MGMT and MLH1 promoter methylation, which are not included here. We have a range of genomic tests we offer for cancer patients, thereby providing comprehensive solutions to our referrers. So from left to right, you will see tests that increase in size and complexity, all the way from an automated 50-gene amplicon-based panel, which has a limited use in our lab, to several larger custom and off-the-shelf hybrid capture-based panels as well. We use multiple platforms for sequencing and automation, including those from Illumina, Thermo Fisher, and MGI. We currently offer many custom medium-sized panels using twist capture chemistry and have off-the-shelf CGP panels from Illumina, including TSO500 and TSO500 HRD, both NATA and CAP accredited. But today we will focus on this new fully customized CGP offering called Benchmark 659. For those who may not be totally aware of the concept, comprehensive genomic profiling allows simultaneous analysis of a large number of genes. No set number really, but say about 350 genes and above would qualify for a CGP tag. CGP allows for a broad coverage and reduced need for multiple tests. CGP plays a crucial role in oncology by facilitating targeted therapy selection and improving diagnosis and prognosis. CGP is now considered a vital tool in biomarker-driven treatment decisions and the potential for expanding research and drug development. So while there are several commercial CGP tests out there, and we have been using some as well, After sequencing thousands of patient samples over the last few years and gaining substantial perspective, we concluded that actually it's time we fulfill the gaps and the challenges we and therefore our clinicians and patients are facing. So we zeroed in on four factors of utmost importance, which we wanted in the new assay, and so worked on incorporating those in the benchmark. So we wanted to work on developing an assay focusing on its accuracy, inclusivity, flexibility, and affordability. I will elaborate on the last three factors in more details, and accuracy will be covered by Lynn later in the talk. Too good to be true? Well, when we set out to work on this project more than 1.5 years ago, we had doubts too. Technical know-how was not a problem, but more challenging was the associated costs, scalability factors, logistical hurdles, etc. We wanted to introduce an affordable test without compromising on standards, and that was the main challenge. We abandoned our plans for some time after commencing, but we eventually got there with the support and understanding of our industry partners. One thing which we never compromised on was the accuracy and reliability of the test. And so huge effort has gone in to make sure that the assay is clinical grade and performs as per our expectations. Benchmark 659 was developed out of necessity to address the limitations of the current off-the-shelf CGP panels. It's definitely not just a large collection of genes for no reason, but one that was born out of consideration for everyday clinical relevance in our work. Too big a panel becomes costly and burdensome, and too small can fall short in clinical use. Hence, it was an intricate balance between cost, panel contents, and purpose. One may say, why not just do whole exome sequencing? And some probably do already. But in our experience, whole exome sequencing is far away from entering routine clinical genomics lab for now. It's just not practical in any way. So here I'm sharing with you the detection capabilities of Benchmark, which are immense. It incorporates UMI calling, and we have validated it to an average depth of a minimum average depth of coverage of about 700 to 800 times per sample post UMI, and raw coverage of around a minimum of 2000 times, and close to perfect uniformity of coverage is what we are getting. Turnaround time is like what we have already been providing for our other CGP panels for last 2.5 years. That is within 14 working days after sample is received in our lab. The assay incorporates 659 genes with 624 earmarked for detection of small variants, copy number change, splice site variants, etc. We have included 111 very carefully selected RNA genes for fusion calling. And as this assay can detect novel fusion partners, it offers a great coverage of almost all clinically relevant fusion transcripts, both for therapeutic and diagnostic purposes. We strongly believe that using DNA-only workflow for fusion detection does not work as well and has limitations outside basic use. And we have always found that extracting RNA and using it for fusion detection is the best way to go, even for FFP tissue. We already have experience of running a 500 gene fusion panel on RNA for the last few years, and it has done exceptionally well for us. Other benchmark features include detection of complex biomarkers like TMB, MSI, and mutational signatures. At the end, I'll also explain as to why we included probes in our panel design to enable us detect chromosome arm level copy number changes or tumor aneuploidy. And considering what this test covers and the panel size, the price point for Benchmark is immensely satisfying for us. So let's talk about the affordability factor. The cost of CGP testing in Australia remains high, which poses challenges for accessibility. The financial implications of costly CGP testing are immense on patients and families. Currently, we are reliant on funded clinical trials, and that is well and good, but how long can that continue? And what happens after that? Then there are other factors that make affordable CGP with quicker turnaround time and necessity for cancer patients. So while this was immensely challenging for us to achieve, and we came close to abandoning the idea because of this cost factor, We had a figure in mind when we started this project, and we knew there is absolutely no point releasing a panel that is not generally affordable for wider use. At $1,300, which is almost half of what typical commercial CGP tests are currently available for in Australia, it opens new opportunities for patients' access to CGP. We hope this brings us one step closer to our efforts towards equity of access to CGP for cancer patients as well. There are many scenarios, some of which I will highlight soon, where we are forced to using 2 or even 3 different tests for answers. Affordable CGP will help us with this issue as well. Another major advantage of affordability in genomic testing is that because more patients can be tested, it allows for more comprehensive biomarker screening for patients. Let's take microsatellite instability as an example. With FDA approval in place and clinical trials and compassionate access available for immunotherapy in Australia, for any MSI high tumor, testing reflexly for MSI in every cancer patient's biopsy becomes an attractive option. Currently, this is not happening outside of clinical trials. Screening for MMR deficiency through IHC is currently limited for a few scenarios only. I have highlighted a study where the authors looked at MSI results for more than 26,000 cancer patients of all kinds. They found that nearly 4% of patients had MSI high status. That is nearly 1,000 patients, not a small number. Almost 17% of endometrial cancer patients return MSI high status, with data from other tumor types like upper urinary tract, small bowel, and gastric cancers also showing substantial number of positive cases. This just shows that with equity of access, we could screen we could screen many types of genetic variations reflexly and treat cancer patients so much more effectively. Second, and one of the most important factors is the flexibility component of our ideal CGP assay. Flexibility in updating gene contents in CGP panels is essential to keep pace with therapeutic and diagnostic advancements. We plan to revise and update the gene contents for benchmark 659 on a yearly cycle, ensuring it remains relevant and effective in addressing evolving clinical needs. The rapid emergence of new biomarkers for clinical trials necessitates regular updates to genomic panels. The illustration on the right highlights the number of trials completed in 2024 for different areas of medicine, with oncology taking top spot, around 1,466 trials completed in 2024 itself, which is not a surprise. Currently, we are finding this as a major challenge with CGP panels, which are not amenable to updates, and we are dependent on commercial entities for update cycles, which can be years in waiting. We plan to be in regular touch with oncologists, researchers, and pharma to gather evidence for most important upcoming biomarkers of promise for each update cycle. Here are a couple of good examples, starting with MTAP gene deletion, which has since last year emerged as a target for a number of clinical trials in Australia. While CDKN2A2B, which resides adjacent to MTAP on chromosome 9, and are readily available in most panels, MTAP is not included in all CGP panels. This has created a bit of a roadblock for clinicians and pharma locally. A good example of a gene that needs to be included is missing. We already have it in benchmark, and soon all our smaller pan-cancer panels will get updated as well with MTAP inclusion. MTAP gene deletion is a target for drug development, now reported to be present in almost 15% of cancers. Here I won't go into details as to the mechanism, but simply put, the therapeutic vulnerabilities created by MTAP deletion presents unique opportunities for targeted treatment strategies. Another good example of importance of regular assay updates is the emergence of LTK fusions as an actionable target with ALK inhibitors in lung adenocarcinoma patients. These are biomarkers that have immediate clinical impact, but it's not easy to screen for them when they cannot be included in our tests promptly. So this trend with newer clinically significant biomarkers emerging on the scene at fairly regular intervals will continue and even gather pace given the speed of drug development globally. And genomic labs need to be strategically prepared for this in terms of ease of testing for emerging biomarkers. The third factor, which is inclusivity, is equally important for a busy genomic lab dealing with high volume of cancer biopsies. A substantial component of our work now is also of diagnostic nature, which is more challenging but exciting as well. We require our CGP panel to go much further than just therapeutic use. Each diagnostic case is unique and brings a unique set of challenges for the team. And we look at many factors, all the way from sample size, quality, tissue and tumor adequacy, sequencing data quality, and the cohort of variants detected. With these diagnostic cases, we must put all these various factors together and correlate the genomic findings with histological impression, and then produce an integrated diagnosis for our efforts. CGP assays must address both therapeutic and diagnostic needs, as histological examination alone is not sufficient most of the time. Let me show you what I mean. Just like rapid emergence of newer biomarkers for treatment and clinical trials, the WHO and other international guidelines for tumor diagnosis and classification are rapidly incorporating molecular characteristics together with histology for accurate diagnoses. While this is all well and good, we only get a small fragment of tissue in many cases, and all tests need to be performed on this small biopsy. I've given you some real examples here. As you can see, some of these tumors that we get are really, really tiny, and we only have one chance to sequence them. So our efforts are now hugely directed to minimize the number of different tests we need to perform to fulfill the requirements. Here are some broad examples of tumor types where molecular profiling has become hugely influential and need to be considered and integrated with histological impressions to produce final diagnoses. Here is a classical example of what we are dealing with daily. A young patient with sacral mass of unknown origin got biopsied with a small core provided to the histology lab, about 13 by 2 mm in dimension. Histology was not conclusive with a wide differential diagnosis. Several immunostains performed were not helpful. There was an initial suggestion that this might be a sarcoma, so FISH testing on 2 probes was performed to rule out Ewing sarcoma and synovial sarcoma. Both were negative. Keep in mind, tissue is being exhausted in the process. So finally, we were contacted with questions from clinicians, and these are very typical questions that we get asked on a daily basis. Which test would be best? Young patient with anxious family. We want to cast our net wide with both diagnostic and therapeutic tests. Would performing a CGP panel cover the scope of the task? If not, then what tests? Is there a Medicare item available to cover the cost? If not, then what cost? We want the results as soon as possible. Now, we had very limited tissue with the possibility of having one go, which presents as a major challenge in this case as well. So in this case, 2 separate NGS tests were performed. A CGP panel to cover possible DNA changes for diagnosis and for possible therapeutic targets, and a separate comprehensive fusion panel to cover the possibility of this being a sarcoma. The commercial CGP panel failed to detect any actionable biomarkers. A dedicated fusion panel was performed, which detected the HEY1-NCOA2 fusion, which is diagnostic of mesenchymal chondrosarcoma. This fusion gene is not included in the commercial CGP panel. So now we have the power to do all of this together in one assay and save a lot of time, hassles, tissue, and also cost for the patient and the clinicians alike. A particularly important example of molecular histopathological integration is that of brain tumors. The 2021 WHO CNS tumor classification is a paradigm shift towards heavy integration of molecular findings for almost each type of tumor, both adult and pediatric. For adequate screening of these tumors, the requirements now is to test for not only mutations, but there is critical importance for copy number changes in some genes. Many fusions are diagnostic for entities, as well as changes in copies of whole chromosome arms are required. Showing you another example. 45-year-old with long-term treatment refractory epilepsy, imaging showing possible type 2 focal cortical dysplasia. Stereotactic craniotomy for excision was performed. Histology came back as low-grade glial tumor. They suspected that this could be an angiocentric glioma, but the diagnosis is quite challenging with these cases and needs molecular profiling for conclusive evidence. So looking at this case, obviously we have to look at both DNA and RNA profiling, because this may not be angiocentric glioma. So the fusion panel picked up a MYB-KQI fusion, which is diagnostic of angiocentric glioma as per WHO guidelines. Here is another example. Young female patient with a cerebral brain tumor, histology suggestive of low-grade glioma, with several differential diagnoses entertained. Clinician and family wanted comprehensive genomic profiling to help with accurate diagnoses and any potential therapeutic biomarkers. So adequate screening here would require not only mutational profile, but several fusions need to be ruled out, as well as changes within whole chromosome arms need to be examined. So eventually 3 different tests were performed. We performed a commercial CGP panel, and then we also performed our custom neuro-oncology panel, as well as a comprehensive fusion panel was done. So what are we dealing with here in terms of differentials? The entities that I have presented here in front of you are all part of a differential diagnosis here. They're based on what we knew from clinical notes, histology reports, and slides that we received. Like many other brain tumor biopsies we receive every day, this will be an explorative exercise with many differential diagnoses need to be entertained. I won't go into details, but all of these differentials are connected to a unique molecular profile, which we can actually screen in our lab now through one test. So anyway, commercial CGP panel showed P10 mutation, which does not provide all the answers. We did our OncoStrands comprehensive fusion panel. No fusions were detected, but it helped us rule out many low-grade brain tumors which are associated with diagnostic fusions. We performed the OncoStrand neuro-oncology panel, which showed chromosome 7 gain and chromosome 10 loss. The panel also picked up the P10 mutation, which was detected by the CGP panel. So in conclusion, we know that this is a high-grade lesion, and the changes we saw in the neuro-oncology panel are diagnostic of glioblastoma, which is actually a high-grade lesion, not a low-grade lesion, which was initially the impression on histology. So we now have the ability to do one panel, which is benchmark panel, which will cover all of these changes in one go. Let's look at sarcomas now. The soft tissue tumors and sarcoma entities I have shared with you have all been diagnosed in our lab with our comprehensive hybrid capture fusion panel in the past and are now included in the benchmark. However, the common theme to note here is that, like the initial example I was sharing with you on mesenchymal chondrosarcoma, many of these samples we receive with question mark sarcoma or soft tissue lesions still need to be screened broadly for both DNA and RNA targets. And that's where benchmark will come very handy. I can't go ahead without mentioning that hybrid capture CGP panels like the benchmark are capable of detecting novel fusion partners, which is such an important point to make because of the promiscuous nature of some of the sarcoma genes, where one gene can partner with many fusion partners. The diagnosis changes depending on which partner gene is detected. Amplicon-based assays don't have the ability to detect novel fusion, and so in our view are useless for this exercise. I have provided 2 examples on the screen. One is for NTRK123 genes, which now have reported about 80 possible fusion partner genes for these 3. And the one on the left is about EWSR1 gene, which can partner with many different types of genes with different diagnoses each time it partners with a different gene, and thereby different treatment outcomes for the patient based on those fusion partners. Talking about sarcomas, we get several uterine soft tissue tumor cases sent to us for accurate diagnoses. Some of the ones we have seen are on the screen. It's important to do this as the fusion detected is not only diagnostic, but also provides prognostic clues in some cases, because depending on the fusion that we detect, it could be a low-grade or high-grade endometrial stromal sarcoma or other types of sarcomas. It's also important to add here that we have also included DNA and RNA changes for many other types of tumors, which the clinicians had asked us to investigate. Good examples are vascular tumors, salivary gland, and renal tumors. We have included biomarkers to help establish accurate diagnoses for some low-grade spindle cell lesions, which again need both DNA and RNA profiling simultaneously. A good example is a cellular desmoid tumor. They can look like sarcomas on histology, but there are characteristic mutations for these lesions, which if detected, are strongly supportive of the benign diagnoses. Moving on to other useful features of benchmark, mutational signatures are included. And I'll let Lynn elaborate on this a bit more, as this is her area of expertise. Regarding TMB calling, all I would say is that benchmark panel size is well suited to calling TMB accurately. There is some good published data out there that proves that the TMB accuracy calling decreases below a certain TMB accuracy calling decreases below a certain size of CGP panel. So it's a good thing in favor of benchmark that it is towards the higher end of the panel size. A very exciting feature of benchmark, which initially we will be using in limited settings, is the ability to analyze tumor aneuploidy or somatic copy number alterations at whole chromosome arm level. But we will keep developing utilities for this feature as we go. In certain tumors like brain tumors, this has already been formally embedded in WHO and international guidelines as a requirement for correct diagnoses, and many other tumor types like melanoma, there is enough evidence of these changes, which are being exploited now. Aneuploidy is also of prognostic importance. Now there is interesting data emerging of therapeutic relevance in these changes are associated with. Detecting aneuploidies present unique research opportunities in cancer treatment. There is emerging evidence, like the two studies I have shared with you here, that highlight the potential for developing new therapies and optimizing existing treatment for highly aneuploid tumors, emphasizing the importance of ongoing research. Studies are now showing that in high tumor aneuploidy cases, there is a synergistic effect of combining immunotherapy with radiation therapy, now emerging as a promising approach for improving patient survival rates. In this study, the authors demonstrate that highly aneuploid tumors can block recruitment of immune cells, rendering immunotherapy less effective. However, with the addition of radiotherapy, it facilitates recruitment of CD8 positive T cells, thereby improving treatment. So we are planning to offer this panel to researchers with interest in this area who are in need for combined CGP and tumor aneuploidy data. Before I finish, I would like to emphasize that Benchmark 659 represents a collaborative effort with industry, showcasing the importance of partnership in advancing genomic medicine. Our clinical and technical input, combined with collective efforts from our industry partners, is an excellent example of how we can contribute to the development of innovative genomic solutions. We would particularly like to thank Decode Science, our supplier for many of the benchmark test components, for being flexible and for working so hard during the validation process. It would be worth mentioning here that an extremely demanding and dynamic test like benchmark requires a robust but flexible secondary and tertiary analysis tool for precise data handling as well. We found a good solution in Franklin. We would like to thank the entire Genox team behind Franklin for working extremely hard with us for more than 6 months to get the bioinformatics pipelines and report templates to our satisfaction. It was certainly no small feat. And finally, our partnership with Twist Bioscience has already led to the development of several custom assays. All the custom Twist assays we have previously introduced and got NATA and CAP accredited and now clinically used are superb tests with a large referral base. So we are actually using the whole workflow from Twist for their target enrichment, including library prep and hybridization reagents as well for these assays. The Twist team has been a great support over the years. Biggest factor of all, they understand our clinical needs and limitations and challenges of a clinical genomic lab in general. So huge thanks to them for all the support in getting Benchmark over the line as well. Now that was my last slide, and I'll pass it on to Lynn to continue from here. Thank you.
Thanks, Vivek. Okay, now I'll talk about the validation of this rather complex and large test. As with all of our tests at LifeStrands, we validate them according to NPAT guidelines. And in this case, because it is such a comprehensive test, even more comprehensive than usual comprehensive genomic profiling, we wanted to do a very complete job. So we've selected over 160 samples that we've tested in-house with various other tests. And that includes mainly clinical samples, but also some reference standards. All of the clinical samples that we have used in this validation were previously tested with our other accredited tests, mainly the Twist panels, but some other commercial CGP tests as well. Because the CGP test that we also used prior to this at LifeStrands is probably the closest in breadth of genomic coverage to the benchmark panel. Most of those cases, I think, are of the highest validation utility, but we did need to backfill the fusion cases from our dedicated fusion test because the CGP, the commercial CGP panel has a very limited coverage of those fusions. So it's lucky, I think, that we've been offering such a broad selection of tests so that we have samples that we can use to validate this new, very, very comprehensive panel. So the plan, obviously, with any validation is to look at accuracy, limit of detection, repeatability and reproducibility, robustness, and then finally, which is not part of the Twist panel itself, but looking at the annotation of the data from the panel, making sure that we have an appropriate analysis pipeline, and that leads into an efficient way to report these clinically. Next. We're also fortunate, I think, because we've had so many samples sent to us of varying cancer types. We have all the usual suspects, lung cancer, breast cancer, colon cancer, but then a lot of sarcomas and CNS tumors and some other more rare types of cancer. So I think we're able to get a nice broad coverage across all of those different cancer types. So as Vivek nicely explained in the 3 clinical cases that he was talking about previously, where we had to perform multiple tests to get the answer for the patient, the same thing is happening with our validation. So I've just shown you on the left side the tests that we have been offering, or accredited tests, and the kinds of variants that each one can detect, and then how that we can use, well, we need to use all of those different samples and different tests to validate this panel, because all of those different tests offer a slightly different piece of the picture. So our very small panel, just the 50 gene hotspot panel, will do limited SNVs, indels, copy numbers, and some fusions, whereas the extended panel will offer MSI as well. The neuro-oncology panel can offer some TMB and chromosome arm level copy number changes. And then the comprehensive HRD panel can offer some fusions as well as TMB and MSI. So we needed all of these really samples that were tested on all of these tests to validate the benchmark to the standard that we wanted to do. Next. So in the samples that we gathered to test, we ended up with, and we're still, I don't think we've completely finished our validation. There are still some things that we'd like to check out. But to date, we've done 113 small variants, including single and multi-nucleotide variants, small indels, frame shifts, non-stops, all of those things, where small variants is anything under 50 base pairs in length. 37 different copy number variants, and that includes both loss and even heterozygous or homozygous loss and gain. Microsatellite instability with a pretty broad range of the percentage of unstable sites that we've been able to measure. Tumor mutation burden, 40 cases, again, with a very wide range. Large genomic rearrangements. This is one of the things that I thought actually was most exciting. These are insertions and deletions that are greater than 100 base pairs in length. And finally, 52 fusions or splice variants, if you want to consider them intragenic fusion. And this, as Vivek noted, is a broad variety of partners because we can do novel fusion discovery, as well as a small number of known clinically significant splice variants. And I'll just tell you right now, sorry, can you just go back one slide? All of these validated very nicely. There has been a lot of back and forth, I guess, on some of the more complex things like TMB, because it is a complex biomarker. It's predicated on getting everything else right underneath it. There's been a little bit of back and forth on the tertiary analysis, making sure that we can get concordant numbers with Franklin. But this is not a validation of the Franklin platform, this is validation of the Twist panel. So just keep in mind that I'm trying to show you Twist data, not necessarily validating Franklin, even though it's been working very, very well. I think it's important just to make the distinction that you can't do one without the other, but mainly this is a talk about the Twist panel mainly. So yeah, we've been very happy that everything has worked out. Maybe just a couple little cases, especially with the TMB that we're sorting out, but we have no worry that we'll be able to get everything as accurate and exact as we'd like it to be. Okay, now you can go ahead. Thanks. I'm still learning the Franklin platform. It's quite complex, especially the back end. So I'm just showing you an example of one case here, just to show you what the coverage and the QC metrics look like. This is a representative case, though, of everything that we've seen so far. And you can see with the percent of the target covered at various steps, there's very good uniformity. Everything is getting covered thoroughly and nicely. We've gotten a rather startlingly huge number of reads and a very low percent of duplicates. This sample that I'm showing you here actually has a mean coverage of almost 1,600, which is a lot higher than we need. And we actually have been trying to get lower coverage because we don't need this much. But we can't because the flow cell keeps overperforming and keeps giving us more reads than we actually ask for, which is a good problem to have. But I would like to spend a little bit more time just seeing just how low we can go, and maybe we can actually fit more samples on a flow cell, possibly even drop the cost further. Another metric that I wanted to point out here is that the percentage of bases in this panel that are not covered is less than 1%, so 0.43 in this particular case, which is more than fine. That's an incredibly small number, and obviously most of those bases are not going to be in the regions that are the most clinically interesting. So we're very, very happy with the QC metrics of the panel so far. Next, Vivek. So just looking at accuracy and limit of detection, we used the Horizon HD729 TrueQ 5% tier reference standard. So this is a useful one because it has a lot of variants that are right around what we expect to be our limit of detection, and in fact what is set as our LOD for all of our other Twist panels, which is 5% variant allele frequency. So I've shown that in that red dotted line and plot on the right. And then I've shown you box plots for each of the variants to show you just how tight and accurate this panel is. The blue, or the teal dot rather, is the variant allele frequency that we expect from the standard. And these all look amazing and super, super tight. And even below 5%, we're still finding variants. So this is very encouraging that we can set our limited detection at 5%, which I think is usually sufficient for tumor cellularity of 10% or greater. So that should be very, very good for every case that that we plan to test. Next, Vivek. Okay, so this is the exciting part for me. I haven't spent a lot of time looking at exon level copy number variants, so I was really interested to see how well the Twist panel would perform on this. We used a TheraSeq large genomic rearrangement somatic mutation mix that they've provided with variants that are at least 100 base pairs in length and sometimes much longer. And you can see here the allele frequency that is expected is about 10%, which is pretty low, I think, for looking at a large genomic rearrangement. And I can't show you all of these, obviously, but I'll let you know that in the Twist data, all of these were present and visible. So we were able to find all of them. And I'll just show you one example next. You can go forward one slide. So here is a BRCA1 100 base pair deletion in this exon. I can't remember which exon it is, that was present at about 10%. We were able to find it at about 6% variant allele frequency. And I've just shown you two different ways of visualizing this. So at the top, you may have to squint a little bit, but if you can see that magenta box that I've drawn, if you look at the left side and then at the right side, you can see how the coverage drops off. And you can kind of see that cliff of coverage on the left and right side where that deletion, yes, thank you, is occurring. And then if you look at the bottom with the stripy colored sections, these are the soft clipped bases of the reads. So these bits of the reads don't align to the reference genome where you expect them to, and that's because this deletion is present. So if you take that soft clip part of the read and you compare it to the rest of the genome to see where it might map, it actually maps to the end of this exonic region, which is shown in the black box at the bottom right. So this is a very elegant example of this 100 base pair deletion being picked up very nicely, very clearly. The Twist panel had absolutely no problem finding this, even at, I think, what is quite a low variant allele frequency. So this was super exciting and very encouraging. Next, Vivek. Yeah, so if I could spend a lot of time talking about fusions, I think this is something that is very important and becoming even more important as we learn more about cancer and driving events and also targeted therapies. So this is a list of the fusions that we've been able to find in our validation, including exon 14 skipping, which is a splice transcript. So this is great. These were all concordant. And if we go forward, I'll show you one case in particular that was very exciting for us. So this is a sample that we tested on our comprehensive fusion panel, and it actually struggled to find it. And this is just to show the sensitivity of the benchmark panel. Even though there were only 7 supporting reads, it turned up quite clearly. So this is a fusion with MEF6 and PHF1. You can see some Franklin eye candy with the fusion displayed, showing exon 4 mapping to exon 2 of PHF1. This is a tumor purity of 30%, which is pretty low. Next slide. And this is just another way to visualize it, just to show you some actual Twist data. Again, I'm showing you the MEF6 breakpoint on the left panel in IGB and the PHF1 breakpoint in the right panel. And again, you can see the soft clipped reads in MEF6, which map to PHF1, exactly where the breakpoint is. And next slide. If you scroll all the way down, there was very high coverage over PHF1. You can find the same soft clip bases on PHF1 mapping to the breakpoint in MEF6. So this is a very confidently called fusion, even though there's not many reads supporting it. It's actually quite clear. So this was gave us a lot of confidence in the RNA component of the benchmark. Next. Here's another example of the high sensitivity in variant detection. So this is a case where we had exon 14 skipping, and we actually found the evidence of the skipping variants in both the DNA and the RNA. So if you look at the middle section of the page with the Franklin variant calls, the top call is the DNA splice donor site deletion, which was called at a variant allele frequency of 9% in a tumor with purity of 10%. You can see this was a core biopsy, only 10 by 2 millimeters, so very tiny. And then the MET exon skipping event was called as the fusion in that second bar with supporting reads of 790 reads, which is quite high, very confident. Next slide. And some more Franklin eye candy. You can see here the DNA variant with that C in the blue circle in the middle. That's where the deletion starts. Just to give you the context, and if you just follow that coding region with the C pro glue, you can see where the splice site is happening in that exon 13. And then next slide. Here's the IGV visualization of it. That's the 8 base pair deletion across that splice site, which is abbreviating the splice site so that the exon is being skipped and causing that skipped exon transcript, which I don't think I showed you, but here's the Franklin version of it, just showing exon 13 being joined to exon 15 in MET. Vivek talked a lot about MTAP and how MTAP deletions are very clinically significant at this point. And there's only, I think, to my knowledge anyway, one other CGP panel that offers actual MTAP coverage. A lot of other panels are calling MTAP deletions as a surrogate of the CDKN2A and 2B deletion, which isn't always the case. Maybe it is most of the time, but definitely they'll be wrong if you don't always look at MTAP directly. So it's great to see this being covered in an affordable panel here. I'm showing you one example where all 3 genes are deleted, or at least partly deleted. So you can see in the Franklin viewer, the MTAP, sorry, just go back one. The MTAP deletion starting on the left side with CDKN2A, the next gene, and then CDKN2B being partly deleted on the right side. And then I'll show you the actual data next. This is just showing you that it is likely a homozygous deletion with 0 copies called. And then you may have to squint a little bit, but if you can see in that magenta box that I've put on the IGV screenshot, the coverage across MTAP, which is quite low. Those little histograms show low coverage, and then low again across CDKN2A. And then you can see halfway into CDKN2B, where I put that little magenta arrow, suddenly the coverage pops up again, so that you can see this gene is not completely deleted, it's only partially deleted. But this is amazing that we can actually just eyeball this in IGV and just look at the reads. The Twist panel, I think, is very consistent with coverage, and it makes it possible to just eyeball these sometimes and see the relative changes in coverage leading to that deletion call. Okay, next slide. So the complex biomarkers, I think, are a really interesting and important aspect of comprehensive genomic profiling. It's kind of a complex interplay, I think, between the mutagenic processes that give rise to the tumor and drive the cancer, and then the genetic artifacts that you see as a result. So a lot of panels can call high TMB, but they don't necessarily tell you what's causing that high TMB. So it could be DNA repair deficiency, particularly in mismatch repair, which could lead to microsatellite instability, or there could be DNA polymerase mutation, which causes ultramutation. Without actually looking at the mutational signature, you don't know what is causing that, except in the case of microsatellite instability. That's a fairly well characterized one. So the ability to call these complex biomarkers and figure out possibly what is actually causing that mutagenic process, and give us a lot of information about what therapies the clinician might want to decide for their patient, or sometimes it can inform the tissue of origin. So the benchmark panel allows us to perform this signature calling, as well as finding the other complex biomarkers, just TMB and MSI. So next slide. So here I'm just showing you a very, what looks like a very simple plot of the tumor mutation burden correlation between other panels where we called TMB and the benchmark panel. And you can see that the correlation coefficient is extremely high. It's effectively 1. It looks a little fuzzy because I'm showing you both of these on a log scale, and that just makes it look like there's a lot of noise at the very low end. But because we did have such a high range of TMB, it's much easier to visualize that using a log scale. So this was very encouraging. I've put the dotted lines at TMB levels of 10 and 20. I think some people are still trying to decide if they think TMB high is over 20 or over 10. Maybe that's a little bit dependent on the panel that's being used to call that, but effectively, the number that we're getting for TMB is very highly correlated with what we expect. So that's good. Next slide. And then the same plot for microsatellite instability. Again, I'm visualizing this on a log scale just to show you the very broad range in percent unstable sites. So the dotted lines are at about 20% unstable sites, which is around the limits of what we consider MSI stable and MSI unstable. And again, the correlation coefficient was extremely high, not as perfect as it could have been, I guess, like the TMB, but we need to remember that we're actually comparing slightly different things here. So the benchmark usable MSI sites is about 200. And with the other panels, we've only been able to call MSI off of 120 sites. So probably the benchmark is actually even a little bit more accurate, I think, because it has broader coverage of the microsatellites. So that was great to see as well. Next. And then I'll just quickly go through this just to show you a nice example of a mutational signature. It's not a very interesting signature because you can see in this case, the colon adenocarcinoma, it's MSI high, it's TMB high. There's an MSH2 truncating variant. So the driving event is pretty clear. But next slide, if we look at the signatures, looking at just the single base substitution signatures, the algorithm will call signatures on everything. It's a bit greedy. So you can see everything reported here, but none of them are very convincing. They all have very low similarities to the known signatures. So there's nothing in this. And then next slide. If we call the indel signatures, you can see here the mismatch repair deficiency signature is dominant and contributes over half of the mutations to all of the indels that are known. So this is just a nice example to show you that we can call the signature and it's actually reflecting the biology underneath. And finally, I think I just want to end on showing you what the report looks like. That is not a real report, obviously, but just to give you an idea of the structure, we have the patient demographic details, specimen information at the top, any relevant information that we can get from other test results, for example, the histopathology report that accompanies the specimen. And then the really nice thing about Franklin is that we have this very succinct summary of findings. So we know straight up there are 3 variants that were called that were significant. We know it's MSI stable, TMB low. Mutational signatures were not called because the TMB was low. There's nothing to call a signature on. And there are 4 clinical trials that were found. Below that, you can see the variants that were called. So this is the MET exon skipping variant and then the MET fusion or exon skipping in the transcript. Also a BRAF, and then the clinical significance of each of those variants is displayed here so the clinician knows right away what the importance of that specific variant actually is. Next. So below that, it's just as important to say what we didn't find as it is to say what we did find. So here we've highlighted the significant negative findings so the clinician knows that we did actually look at these and there was nothing there to report. No pathogenic mutations in the usual suspects that are important in this type of cancer. Below that, the clinical interpretation of each of the biomarkers. So MSI stable, but the clinical significance of that is in the context of this tumor type. Again, the TMB low and what the significance is of that. Next. And then a description of all of the variants. I'm just showing you one of them here, the MET exon 14 DNA splice site variant leading to exon 14 skipping. Description of the gene, why it's important in this cancer type, a description of the variant, and then therapeutic evidence of whether or not this variant leads to responsiveness to certain drugs or resistance to those drugs or whether it's diagnostically interesting. Next. And then below that's a description of the clinical trials that we were able to find. We do limit this to Australia and the patient's actual city if possible. And then we do report the tier 3 variants of uncertain significance because these could be important later when they become less uncertain. So those are there for future interest.
Thank you both so much for your time and the amazing presentation. That does conclude the end of today's webinar.